Hello friends, this is Supriya. Welcome back to my channel Biology Reader. Today in this video we will learn the mechanism of enzyme action or how the enzyme works. So friends, let's get started. Before proceeding to the mechanism of enzyme action, we must know the basic terms of the enzymology. Enzymes are the three-dimensional nitrogenous organic compounds produced by the living plant and animal cells. They are proteinaceous in nature and having high molecular mass. They participate in various biochemical reactions to increase the reaction rate. Enzymes are never used up in the reaction, they just do their task. It means they convert substrate into a desired product and then just become free to catalyze another reaction. They can catalyze both catabolic and anabolic reactions. In catabolic reactions, a complex substrate is degraded into simpler products, whereas in anabolic reactions, a group of simple organic compounds form a complex product. Then we need to know what is enzyme action. Friends, enzyme action is just the way enzyme works or how it catalyzes the reaction. Due to this, enzyme action is also called enzyme catalysis. Enzymes perform a conversion of substrate into products by lowering the activation energy, which we will further discuss in this video. Sucrase is an enzyme that converts sucrose into glucose and fructose. Then substrates are the reactant molecules that first react or bind to the active site of an enzyme to form enzyme-substrate complex. The active site of an enzyme is specific for specific substrate. Therefore, an active site provides specific binding between the substrate and the enzyme. After that, products are the molecules that are formed at the end of chemical reaction by the conformational changes in the enzyme-substrate complex. Now we will discuss how the enzyme works. Friends, we must have heard that the enzyme functions by lowering the activation energy to convert substrate into products at a faster rate. But do you know how this activation energy is getting reduced? Well, if you don't know, then look into this graph. Here this vertical arrow represents the free energy and this horizontal arrow represents the reaction path. If a substrate is converted into a product without the involvement of an enzyme, we obtain a curve like this. Or we can say that in the uncatalyzed reaction or the reaction without the involvement of an enzyme, a substrate requires more energy to get into the transition state. So in the uncatalyzed reaction, the activation energy or the energy for the substrate to get into the transition state is very high. But in the enzyme catalyzed reaction, the activation energy reduces. Because the binding of substrate and enzyme releases some sort of energy, what we call the binding energy. Therefore, reaction will progress with or without the participation of enzymes. But enzymes speed up the reaction rate thousands of times than the uncatalyzed reaction. Now transition state is the intermediate stage between the substrate and the product. Substrates reach this transition state to actively form products, and this is the overall energy released during the reaction. So through this graph we can conclude that enzymes reduce the activation energy. They function as biocatalysts to accelerate the conversion of reactants into products. And last but not the least, enzymes increase the reaction rate, but they do not affect the reaction equilibria. To understand the mechanism of enzyme action, there are two theories. One is Lockin Key model and the other is induced-fit model. Starting with the Lockin Key Theory, it was given by Emil Fischer in the year 1894. According to this theory, a specific substrate fits into the active site of an enzyme like the way a particular key fits into the keyhole of the lock. Then the binding of substrate with an enzyme forms a temporary complex called an enzyme-substrate complex. Here one important thing to note that according to this theory, enzyme doesn't undergo any conformational changes. It is the easiest model to explain the enzyme specificity, but it has many loopholes, like it fails to explain the broad specificity of the enzyme. This model does not explain the mechanism behind the substrate binding with an enzyme, and it does not give any information about the enzyme catalysis or the product formation. Now coming into the induced-fit model, it is widely accepted theory to understand the mechanism of enzyme action. It was given by Daniel Koshland in 1959. And according to this theory, active site is a flexible region which undergoes conformational changes to produce desirable products. Here the substrate initially interact with the enzyme's binding site or the active site and the catalytic site of an enzyme brings some sort of conformational changes in the enzyme that breaks various bonds formed between the two and results in dissociation of products. So friends, this is all for today. To know this topic more in detail, you can visit our official website, that is biologyreader.com. Link is provided in the description box. If you find this lesson useful, do like, comment, share and subscribe my channel, and don't forget to press the bell icon for more videos.